Myself, Dr. Supratik from the Department of Chemistry. Today, I will present on a topic called an integrated approach to identify dual target inhibitors for Nipah virus through drug repurposing. First, we need to know about the Nipah virus. Nipah virus is a highly lethal RNA virus from the family of the Paramoxypheridae with the genus of Enipavirus. According to the CDC, the Nipah virus is classified under the biosafety level 4 pathogen. That means the virus which has the capability to create the pandemic. After the COVID-19, Nipah virus can be a strong candidate for the future pandemic. So therefore, this is one of the most important virus for the drug design and drug discovery. Fruit bats are considered a primary or the natural reservoir for the Nipah virus. Generally, the fruit bats are the reservoir for the Nipah virus. Once they infected the nectar of the dates and the palm trees, contaminates fruits, they directly and indirectly affect animals followed by the human. The major clinical symptoms of the Nipah virus is like subclinical asymptomatic infection to acute respiratory infection followed by the fatal encephalitis. Generally, patient can experience the fever, altered mental state, headache, severe weakness, and at the end the dizziness. In many of the cases, after the seizure and the fatal encephalitis, the patient can have a death. Now, we need to understand the basic reproduction number or R0 value. Generally, the R0 value is the calculated that can be to understand the communicable disease or the spreadability of a virus. If we consider the R0 value for the flu, it's generally 1. On the other hand, if we consider the Omicron or Delta, it's considered like 6 to 7. That means if a patient has a COVID-19, it can spread to 6 to 7 people. On the other hand, if we compare the Nipah strain of 2009 and the Nipah strain of 2022, it's around 0.5. That means one patient can spread the Nipah virus to only half person. So technically, like 2% can spread to one person. On the other hand, if we see the mortality rate, in case of COVID-19, the mortality rate is only 1.01%. That means if 100 people got affected with the COVID-19, only 1.01% will get death. On the other hand, the mortality rate of the Nipah virus is close to 40 to 75%, which is extremely mortal. So you have to understand that if there is a reason of a mutation of those Nipah virus in the bat reservoir, and they can have a much more stronger R0 value, the mortality with this high mortality rate, the virus can be extremely deadly. There is no present medications or USFDA-approved drug or vaccine for the Nipah virus. Ribavirin, chloroquine, or remdesivir, there are some drugs. They are mostly checked over the preclinical studies, but nothing is approved yet. There is a monoclonal antibody M102.4 has passed the phase 1 clinical studies, but again, is not approved yet. In the present study, we have targeted Nipah virus attachment glycoprotein human ephrin B2, which is NIVG, and matrix protein NIVM as dual targets. The major reasons are the following. NIV was found to infect cells through fusion and attachment of glycoprotein by a pH-independent membrane fusion process. Among the process, ephrin B2 is identified as the primary tropism of the Nipah virus, and the role of the glycoprotein in facilitating the virus entry into the host cells through affinity protein-protein interaction is essential to target. Therefore, the Paromix virus matrix is a key target for the antiviral responsible for protein-mediating virion assembly budding from the host cell membranes. That's the reason we are targeting the virus Nipah virus attachment glycoprotein and Nipah matrix protein. Under the adapted scheme for the present study, we have considered the drug bank approved 2,588 molecules for the drug repurposing, which generated the confirmations. On the other side, based on the site map, we have identified the active site of the both proteins, and followed by we have run the XP-based glide docking, admin profiling, molecular dynamic studies, and MMG-based studies, And based on the overall analysis, we have proposed dual target inhibitors for the Nipah virus. This is the docking score for the top 8 identified ligands. All the docking score are extremely high compared to the existing drug molecules for many other diseases. Here is the ADMET profiling for the 5 screen ligands. Except the DB01698, almost all other 4 molecules are extremely safe considering ADMET profiling. This is the results for the 2VSM and the 7SKT, where we have find that there is a very, very small deviation of the RMST over the 500 nanoseconds if we compare along with the apoprotein and the complex ligand. We have not seen more than 1.5 angstrom root mean square deviations for any complex in both proteins. On the other side, we have run molecular mechanics energized combined with the generalized bond and surface area, which is MMGBS study. And here we have find that in case of targets 2VSM and 7SKT, ligand DB01249 and DB0947 demonstrate the most consistently low binding free energy throughout the 500 nanosecond simulation. If we see the results for the 2VSM and 7SKT for the DB01249, again in both cases we have not seen the root mean square deviation more than 0.6 angstrom, and we have seen a very strong binding with the hydrogen bond formation and the water bridge. On the other side, we have also plotted RMSF per residues is typically plotted versus the residue number and can indicate structurally which amino acids in the protein contribute to the most of the molecular motion, where we have seen most of the cases the hydrogen bond and water bridge are the most important one. A similar pattern has been seen in case of our DB09487 for the both proteins. For the free energy landscape, the color gradient represents the frequency or the probability of the protein being in a particular conformation, where the red areas indicating the low probability high energy states and the blue areas indicating the high probability low energy state. The apo form of the 2BSM shows a free energy landscape with the distinct minimum indicating a stable conformation with a low RMST value. On the other side, the protein likely adapt a specific structure or a set of close related structures over the time. On the apo state, 7SKT shows a relatively broad distribution of the conformation with a low RMST value, which indicates a stable conformation throughout the 500 nanoseconds for both ligands for both proteins, comparing to the apo structure. On the other side, for the PCA plot, which suggests that the progression of the frames from the beginning to the end of the simulations, with the starting frames in the purple and ending frames in the yellow. The principal component analysis is the axis along which the data's variance is maximized. In other words, it represents the direction in which the conformational changes of the protein are the greatest. If frames are clustered close to the PC1, it means the conformational changes are predominant along with the motion captured by the PC1. For the protein ligand complex, this could mean the most of the motion due to the binding occurs along the motion described by the PC1, which you can easily see from the color deviation. We have also performed the defined secondary structure protein analysis, which represents the structural changes of the two different proteins of the Nipah virus visualized at different time points from 0 nanoseconds, 250 nanoseconds, and 500 nanoseconds of the MD studies. Over the 500 nanosecond simulation time, the apo form of the attached glycoprotein maintains its overall secondary structure throughout the 500 nanoseconds. On the other side, when we have a ligand complex, we have seen very much deviation in the structural change to show the binding score. Overall studies suggested that we have found that dual virtual screening inhibitors, which are the irinotecan and iodoxinol, which can be extremely potent for both targets. On the other side, routine or lacitol can be a very good target for the matrix protein. Considering the interpretations, we have understand that four molecules has a sugar-like structure which have glycomimetic properties to interfere the glycosylation process of the Nipah virus to kill those viruses. On the other hand, the dual targets has a nucleobase-like structure and amide linkage which can act as a mimic the structure of the natural nucleoside to interfere the replication process of the Nipah virus, as well as the inclusion of the amide linkage in those molecule structure may enhance their binding and activity against the Nipah virus. Overall, based on our study, we have found two molecules which can be extremely potential lead molecule for the Nipah virus. From another study, literature has shown that these molecules are extremely important for the as an inhibitor for the SARS-CoV-2. We are looking for the collaboration to check those drugs in the experimental setup to make this possible to lead molecules as the future drug for the Nipah virus. I want to acknowledge my student Shion Yang, who has performed most of the study, and we have a very fruitful discussion over the time. I want to acknowledge the AC Asymmetric College, our Department of Chemistry and Physics, Dean Chang, Chair Dr. Stokes. I also want to acknowledge US EPA for the federal funding to buy the software from their money. Thank you so much.